Dr. Nicolas Abreu, the diagnosis of an ultra-rare genetic disorder for many of us, brought a long-awaited answer to symptoms that could not be explained. At the same time, it introduced us to a world of rare diseases that many of us had no prior exposure to. Parents of children with rare gene disorders are amongst the most expert individuals in the room at the pediatrician or the school IEP meeting. Yet how the gene disorder occurs can still be confusing. Today, we welcome Dr. Nicolas Abreu, a specialist in adult and pediatric neurogenetics. And I don't know if I'm saying this right, Dr. Abreu serves on the Patient Outreach Committee of the American Society of Gene and Cell Therapy, which is how we have his expertise available here today. He was a peer mentor in high school to classmates who had a range of physical and developmental disabilities, including cerebral palsy, autism, and intellectual disability, which led him to pursue a career in child neurology and neurodevelopmental disabilities. During residency, he encountered families like ours who had been on a long journey for diagnostic answers that resulted in a genetic diagnosis. Today, we are privileged to learn from Dr. Abreu about the topics of genetics, which has come to be a knowledge that we are all grasping to understand better. Figure out how to stop sharing. Here we go. And if you can share, oh good, you can share. Take it away, Dr. Abreu.
Great. Can you hear me okay, Disa?
I can hear you. I see your whole, there we go. I see your presentation. Yep.
That works. Okay. So welcome all, and thank you very much for inviting me, Disa, and the entire ZMYND11 community. I am a child neurologist, neurodevelopmental disability specialist out of NYU, and am not expert in this condition like you all are, but from my position as a neurogenetics doctor, am here to provide a bit of perspective on the genetics of ZMYND11 related what I'm calling neurodevelopmental disorder, about the other names, and also share a bit of information about the parent organization that I volunteer with, ASGCT. So ASGCT offers a speakers bureau, which is how I got connected to you all. And in particular, the ASGCT is the primary professional membership organization for researchers, clinicians, and advocates working in gene and cell therapy, with the goal of advancing knowledge awareness of these therapies for discovery and access to treatments. The Patient Outreach Committee is just one of multiple committees within the overall organization. And we're doing a lot specifically to partner with patient advocacy organizations and directly with affected individuals and their loved ones to improve understanding of not only clinical genetics, but really genetic therapeutics. And if you're interested in learning more, after this talk, there's a QR code to scan to learn about gene therapy basics, and then also in general information about patient education. We also have some short videos about issues in genetic therapies, ranging from how do we measure treatment response using things like biomarkers, how do we make gene therapies or genetic therapies, the complexities about whether or not or how to participate or consenting towards doing one of these clinical trials, and the process and what's involved in genetic testing and how that may inform eligibility or clinical trial design or implementation. So we'll talk a bit about overall strategies in terms of, or overall basics of genetics. We'll talk a bit more about genetics as it applies to ZMYND11 related neurodevelopmental disorder, and talk a bit about the clinical characteristics, understanding that this is really a conversation to spark further dialogue with your healthcare providers, and also to learn more with the other speakers later on during today's session. DNA, or deoxyribonucleic acid, stores our genetic information, and it's located in the nucleus of each of our cells in the forms of chromosomes. Each, uh, each is made up of 2 strands, and it's considered a permanent storage format. Our DNA are arranged into sections known as genes, and those genes encode sequences or the instructions to make proteins. Other sections of the DNA, or non-coding regions, are control when, where, and how much protein is made. And protein is ultimately the thing that does work in our cells. RNA is a copy or is transcribed from DNA and serves as a template for making protein in the end. It's made in the nucleus and travels to the cytoplasm with instructions to make these proteins. However, there are still other types of RNA that can control how proteins are being made, so there are different functions. And RNA overall has a short time frame of living in our cell, or is temporary. And when thinking about how do we treat genetic disorders or common disorders that might have a genetic basis, we have different strategies, either treating DNA or RNA. And at the very end of this talk, we'll talk about how this links up with ZMYND11 related neurodevelopmental disorder. For genetic disorders overall, we can think about gene addition, which is currently being used in clinic for a handful of conditions, where we add in a working copy of the gene that's not functioning normally in a human. And that gene has the instructions for the cell to make more of a specific protein that's needed. In contrast to gene addition, gene silencing, we can add genetic material to prevent or inhibit or tamper down activity that's already present in a cell. Gene silencing often decreases the amount of a specific protein being made in contrast to gene addition, where you're trying to make more of a specific protein. Gene editing can be used for either making more or making less, because you're correcting pieces of DNA by changing or deleting information within someone's genes. And because we can directly edit, we can either turn up or turn down the resulting protein being made from that gene. A third or a fourth approach can be genetically modifying cells outside of a human, which is currently being used for some conditions, requiring removing of patient cells from the body, modifying them outside of the body, and then reintroducing them into the body. And this is currently being used for some neurological conditions as well as for certain types of cancers. The American Society for Gene Cell Therapy provides education to families, industry sponsors, clinicians, and researchers in terms of the complexities of genetic therapy clinical trials, how to move from the early stages of clinical trial design and evaluation, even in the steps where we're trying to see if a new therapy or new genetic therapy could be helpful to certain cells or to animals, what's known as preclinical studies, all the way through to applying for a new drug application to the FDA, to early phase trials, all the way to pivotal trials, and then ultimately, at least in the United States, approval by the FDA for use in clinic. There are also long-term follow-up studies that often need to be done, even if it's approved early. But certainly the goal of any kind of rare genetic disorder, once we know the underlying cause or the genetic change, is to try to think about how could we correct or treat the underlying gene change or genetic problem so that we could have long-lasting improvements or treatments for that given condition. And there are a lot of other cases, both in the neurological space and others, for which gene therapy has moved through each one of these steps. And sometimes it's not as smooth as just one after another, and sometimes can take many years. But ultimately, this is the hope that there are new treatments for ZMYND11 related neurodevelopmental disorders in the future, working between an organization like yourselves, as well as with clinicians and researchers to move this process forward. So I'm using the term ZMYND11 related neurodevelopmental disorder, or ZMYND11 NDD, and it goes by different names depending on who you talk to. I prefer to use the term neurodevelopmental disorder to be more encompassing, but with a focus on the challenges in neurological function and developmental disabilities that is oftentimes seen. But other people use terms like ZMYND11 syndrome, just related to the gene specifically, or to highlight difficulties in cognitive functioning that is very often seen, namely ZMYND11 related intellectual developmental disorder, or just in terms of the time at which the gene was described to be associated with this condition. So it's associated with a certain number in terms of the year at which it was described, otherwise known as intellectual developmental disorder, autosomal dominant 30. For me going forward, I'll use the term ZMYND11 NDD. All of these are the same terms for the same thing. But what is ZMYND11 NDD? It's an autosomal dominant neurodevelopmental disorder due to a pathogenic variant in the gene ZMYND11, characterized by some typical features. That includes early developmental delays, variable intellectual disability, behavioral differences, sometimes seizures, sometimes low muscle tone, sometimes, actually rarely, some skeletal differences, sometimes feeding difficulties, and sometimes differences in facial appearance. That's a lot of information, and we'll break down the first main part of this definition above. What do we mean by saying autosomal dominant neurodevelopmental disorder due to a variant in this gene? Well, here's a depiction of a cell. In the cell, we have our instruction manuals to our bodies or to our cells, known as genes. And those genes are found in the nucleus, again, these X's or chromosomes, and each chromosome is composed of one large continuous DNA molecule. You can see here this big X wrapped up with a single wrapped strand or continuous DNA molecule that forms, when we unravel it, a series of letters. These letters or nucleotides are adenine, thymine, guanine, and cytosine, also known as A, T, C, and G. And so you can think of our DNA is made up of 4 different letters. Just as if there are 26 letters in the English alphabet, there are 4 letters in our genetic code. And the particular sequence or whole segment of DNA that encodes a protein product, it's what's known as a gene. And we have over 20,000 genes in our body. Just so happens is that if you have a particularly significant change in the gene ZMYND11, you can have the neurodevelopmental disorder that we just described. Each gene encodes a protein, and the protein is made up of hundreds or thousands of amino acids. Each oval here is an individual amino acid, and so this string of amino acids makes a protein that does work in our body. How we make protein goes through a standard set of processes in our bodies. So DNA, made up of a string of A, C's, T's, and G's, gets read and gets made into mRNA, or is the template for which we make protein. So DNA gets made into RNA that then gets made into protein. That process of DNA being made into RNA is known as transcription, and then RNA being made into protein is known as translation. And there are different things that help us go from DNA to RNA and RNA to protein. At the very end of each protein is a stop signal to say, hey, stop making protein. And this is what would be considered the full-length protein being made. And if there's a change in the DNA, that could potentially affect the protein being made and how the protein functions. I like to use the example of our genetic information, or genome, as a genetic library. We have 23 chromosomes, or 23 rooms within that genetic library. And within each room, or within each chromosome, there are shelves arranged with a series of books, or books being genes. And so what we can do with genetic testing technologies is read through different sections of that genetic library to look for changes or spelling errors in different books or genes within that genetic library or genome. And we have different ways of looking across that genetic library or genome. You all likely had some experience working with genetic testing, and there are different types of genetic tests that could be associated with identifying ZMYND11 related neurodevelopmental disorder. While light microscopy, or looking under a microscope to look at our chromosomes, has previously been used, right now an old standard genetic test is known as karyotype, where you can see these bands of genetic information. And that looks at the very, very big picture, looking to see if whole sections of DNA are missing or rearranged. That looks at a very, very bird's eye view. And more often than not, microarray is a next more detailed type of study, looking to see if whole shelves of books or whole sections of DNA are missing or copied. This looks at more detailed resolution than a karyotype, but not as detailed as some other types of tests. This can detect sections of that genetic library where whole shelves up to 50 to 100 kilobases, or 50, and that's kind of like the size, the number of letters that are missing, looking for large sections that are missing or copied. And changes, especially deletions, can sometimes be detected on microarray, for which previously or early cases of ZMYND11 related neurodevelopmental were detected. Now, more commonly, we're using something called exome sequencing, which looks letter by letter at individual books or individual genes within our genetic library to look for spelling errors. So we're looking letter by letter, or base by base, or nucleotide by nucleotide. We're looking at a lot of information. And so variation is commonly identified, and variation, or just changes in our DNA, happen in all of us, most of them, most of the time, that are not associated with uh, problems with health or disease. Exome sequencing looks at 1 to 2% of our overall genetic information, primarily focusing on the coding region, so the part of our genetic information that encodes proteins. There's still a lot of other genetic information within our genetic library, within our genome, and that's where genome sequencing starts to look at more information, looking at the majority of variants or changes in our genetic information that can be associated with human disease. There's also more likelihood of finding common genetic changes, or changes that may not be associated with human disease. And overall, the more detailed we look, the more likely we're able to find a genetic disorder, although there are limitations in all of these testing technologies. ZMYND11 related neurodevelopmental disorder has been diagnosed using many of these types of tests, including microarray, exome, and genome sequencing. You may be familiar with reading or receiving a genetic test report, and it can be quite confusing, using a lot of terms that aren't a part of our standard knowledge, and so requires detailed explanations and working through. Here's a case of a previously published individual that I recreated what may have been found on their genetic test report. And so in this particular paper, one individual was found to have a disease-causing change in the ZMYND11 gene, and at the 46th position, where there was a C, there's a T. This was found to be heterozygous and was inherited de novo. The classification from the laboratory was pathogenic. And we'll break down each of these each of these terms in the next slides. So ZMYND11, what is it? It's a protein. The gene is in italics, and the protein is written in regular script. And ZMYND11 is also known as zinc finger mind or MIND, domain containing protein 11. To make things even more confusing, previous literature referred it to as BS69. And the protein is involved in regulating or controlling the creation of RNA. Again, DNA gets made to RNA, which ultimately serves as the template for making protein. So if you control the creation of RNA, then you're controlling the amount of protein being made at the end of the day. How ZMYND11 works is that it recognizes a component of the organizational structures of DNA known as histones and certain areas of the histones to regulate the transcription of DNA to RNA. In particular, here on the right, you can see a picture of DNA depicted as a blue string wrapped around these certain teal or green circles or cylinders. And these cylinders or circles are made up of a series of proteins, and these cylinders are known as histones. They're the things that help package our DNA within those chromosomes. And so ZMYND11 helps repress or tampen down transcription or the creation of RNA by regulating the protein that reads or goes through DNA, known as RNA polymerase II, to make RNA in the end. ZMYND11, the gene, is located on the 10th chromosome. It's located at the very end or near the end of the short arm or P arm of the 10th chromosome. We use certain GPS coordinates when we're looking at the chromosome location. So this is read as 10p15.3. So it's just on the 10th chromosome, the short arm or the P arm, and at the region or subregion 1, subregion 5, and then 0.3 is the specific coordinate within there where the gene is located. We all have 2 copies of our genetic information, 1 copy that we get from mom, 1 copy from dad. And so everyone has 2 copies of the ZMYND11 gene. It just so happens is if you have a variant or gene change on one of those 2 copies, and it's a particularly and causes a negative effect on protein function, someone will develop ZMYND11 related neurodevelopmental disorder. This is also known when you have one of 2 copies being abnormal or having a gene change is known as having a heterozygous variant. Or having 2 different copies of a gene where one of the 2 copies of the gene has the genetic change. What are the consequences of variants in ZMYND11? Well, most of the time, when we're looking at the neurodevelopmental disorder due to changes in the ZMYND gene, they're associated with loss of function or preventing the formation of functional protein. However, there's a question about whether gain of function is actually a possibility as well. Initially, back in 2013, when individuals had a whole section of DNA being missing or deleted, otherwise known as 10p15.3 microdeletion syndrome, they found that individuals had a certain neurodevelopmental disorder. Later, so having a whole region down here being missed. Later, when individual variants or individual spelling errors within the gene caused a loss of function, that later said, hey, you know what, it's not the entire region being deleted, it's really this gene that's most important to causing the neurodevelopmental disorder. In other words, if we knock down 1 copy of the ZMYND11 gene, that will lead to the neurodevelopmental disorder that we see. In this case, for the variant, at the position 46, there was a single nucleotide change. That C dot refers to the reference sequence or the coding DNA in which we found the spelling error. And at that particular position, 46, where normally there's a C, this greater than sign is really a directional sign to say, actually, it's not a C, it's a T in this individual. And so this C was changed to a thymine. That's at the DNA level, but at the protein level, we can see the expected change in the parentheses, or P dot refers to the protein or amino acid sequence. And there was a single amino acid change at the 16th position, where normally there's a glutamine, actually no amino acid was substituted. It created a stop sequence. And unfortunately, when there's a stop sequence early in the protein and nothing else gets read, the body recognizes that very short or truncated protein and destroys it. And so that leads to a loss of function. Most genetic reports will record both the DNA change as well as the expected protein or amino acid change. We talked a bit about the heterozygous, or having one of two copies, being abnormal. And we'll talk about the other words in a little bit. There are multiple types of genetic changes. You can have missense changes, where one letter changes for another, or at the amino acid level, at the protein level, one amino acid gets switched for another, and that can alter the way a protein is made. It can lead to loss of a single letter switch or typo can lead to loss of function, change of function, or gain of function, depending on how that change or what happens in terms of the switch. A nonsense variant is a change where a full stop gets introduced into the genetic code and means that part of the protein after the stop signal will not be made. And then there also can be a frame shift where there's a shift in the genetic code, oftentimes with adding or deleting certain letters or nucleotides within that genetic information or DNA. And oftentimes that leads to the protein being not made properly. There's also changes that can affect how RNA gets processed, known as splice variants, that are beyond the scope of this talk. In our case, where we had one single letter change from a C to a T and introduced a stop codon or a truncating variant, this would be a type of a nonsense variant. How did this happen? So it depends. Oftentimes we need to test parents to determine how this particular change happened. More often than not, it's a new change or spontaneous change that happened during a pregnancy. In this case, you can see two parents, each with two normal copies of a gene. In this case, we'll say ZMYND11. And each parent passes down one copy onto their children. Depending on which copy gets passed down, in all these kids, they'll inherit normal copies of the gene or are unaffected. In this case, in the affected child, there is a new change. And so in the abnormal gene in blue, the child has the genetic change because of this new change not inherited from either parent. In this case, we'd expect both parents to be healthy, and in future children, for them not to have another child with the condition. But even though it's a very, very low likelihood, it's not completely zero due to some nuances in terms of our ability to detect genetic changes in parents. In our case report or our case that we discussed, the inheritance pattern, because parents were included in this in this evaluation, it was a de novo change or not inherited from either parent. There have been cases previously described in ZMYND11 neurodevelopmental disorder where the variant was inherited or was passed down from an affected parent onto their children. This is also true for individuals who are already affected with ZMYND11, that they have the ability to pass on the the variant onto their future offspring. So in this case, an affected parent or affected individual has one abnormal copy of ZMYND11 and one normal copy. And they have a 50% chance of passing on the gene with the variant onto their children. In this case, the other parent doesn't have an abnormal copy or has two normal copies, and so they will always pass down a normal copy. It really is depending on a 50-50 split, which copy from the affected parent gets passed down that then determines whether or not the child is or is not affected. In this case, this represents an autosomal dominant inheritance pattern, or ZMYND11 related neurodevelopmental disorder is an autosomal dominant disorder that follows this inheritance pattern, in large part because having one abnormal copy of the gene causes disease. Another thing that you saw in the report is the classification of the variant. At the very end, the genetic testing laboratory decided to classify this particular spelling change, or at the 46th position in the coding DNA, instead of a C, there's a T, as pathogenic or clearly disease-causing. However, pathogenic just represents one of 5 different possibilities of what variant classification could be. The nuances of individual variant calls can be tricky and sometimes can change either depending on the laboratory that reported the variant or based upon the amount of information available to classify it. So for variants of uncertain significance, which sometimes come up when we do genetic testing, some of those may or may not actually end up being associated with ZMYND11 related neurodevelopmental disorder. And it's important to work with your healthcare providers to know if there's any further evaluations that can be done to help better classify these variants of uncertain significance as either normal or likely benign, or potentially abnormal or disease-causing as likely pathogenic. So how does ZMYND11 related neurodevelopmental disorder present, or what are the different health issues? Early developmental delays, especially speech delay, has been seen in nearly all individuals with the condition. This is reported, these are data from one of the largest papers from 2020 on this condition. 80% had gross motor delay, and this was seen early in development. In this group of over 20 individuals, all had intellectual disability, but range from mild intellectual disability to severe intellectual disability, and that's about one's ability to think, learn, and be independent or perform activities of independent living. Behavioral abnormalities were common, although the types of behavioral abnormalities varied from person to person. And while the majority had some kind of behavioral difficulties, anywhere between 20 to 50% were diagnosed with conditions like attention deficit hyperactivity disorder, autistic features, or aggression. Seizures in this study were reported at 38%, and seizures will be discussed more later in other talks. Low tone or low muscle tone was seen in about half. Differences in the formation of fingers or differences in facial appearance were sometimes seen. Differences in facial appearance were common at 66%, and this short fingers or brachydactyly and changes in the tooth enamel were seen in just a minority of individuals in this study. And feeding difficulties were seen in about 60%. I think in order to conclude in terms of ZMYND11 related neurodevelopmental disorder, it's really important to understand the mechanism to both understand how does ZMYND11 NDD, what kind of clinical characteristics may be seen, as well as how do we actually define treatments. For the majority of cases, these are thought to be, again, related to loss of function, also known as haploinsufficiency, or where one of 2 copies is not working properly or is not making protein, and having the other copy isn't enough to protect against disease. treating loss of function conditions could be done through things like gene addition or gene editing, but would not be appropriate for something like gene silencing. In contrast, other some other variants or some other genetic changes, there's a suggestion that maybe there could be gain of function for which treatment with genetic therapies with things like gene silencing, but not gene addition, may be considered. Regardless, this is complex and certainly are some things that myself as a clinician are thinking about when I review genetic disorders. So with that, we'll stop and we'll open it up for questions.
Thank you, Dr. Abreu. That was a lot, but I think it was the best description I've had so far to really understand how everything works. Does anyone have questions? I know I'm asked quite a few times, and I think you touched on it briefly, that if there is an individual that does have a mutation that is affected, their likelihood of passing on that gene to their kids would be 25 to 50%, or is it like a definite they'll pass it on?
For any affected individual with ZMYND11 related neurodevelopmental disorder, it's 50%.
And anyone is welcome to come off mute and ask questions.
I have a quick question. Hi, my name is Jenny Rankin. Doctor, what do you see as far as having hope that there could be gene therapies in the future? And I hear promising things through AI and being able to generate solutions. Is it realistic for us as parents to think that there could be a gene therapy somewhere on the horizon?
I think there's always room for hope. And I think from what I've seen in clinic for other neurological disorders, we do have gene therapies for those conditions. I think when looking at the spectrum of neurogenetic disorders, treating neurodevelopmental genetic disorders can be hard because one of the main things is where is the window for intervention that could be most helpful. And because we think that this is an issue that this genetic disorder happened from their development in utero and may affect early brain development, even if it doesn't necessarily affect the structure, it will have effects on the function. And so I think for these genetic therapies, even if they are to be delivered, it's when is the right time if we do have these therapies. In contrast, there are other complex genetic neurodevelopmental disorders for which we have maybe not gene therapies, but we have oral therapies or small molecule therapies. And Rett syndrome is a great example of one that recently got FDA approval. And there's a bunch of other conditions for which, even if it's not necessarily cures, thinking about ways to improve quality of life, whether it's through genetic therapies or small molecule therapies, are certainly possible. Because we understand that it's not just while cure is certainly a goal, there are a lot of different ways that a neurodevelopmental disorder can impact day-to-day life, and whether it's behavior, speech and language, motor functioning, there are a lot of different ways that we could use drugs or use treatments to improve quality of life.
Could you give us an example of like some type of treatment that someone with ZMYND11 NDD could use to give them some relief from their symptoms?
So I won't specifically refer to ZMYND11 related NDD, but in other conditions, so we have gene therapy approved for a rare neuromuscular disorder known as spinal muscular atrophy, and this causes, in the severe form, weakness around the time of birth and typically death by 2 years of age related to problems with the nerves, the motor nerves, going from the spine to the arms and legs. And when we give this gene therapy, a normal copy of the gene that's involved in spinal muscular atrophy to babies, through a vein, just like in an infusion center, we see that these children are no longer are actually starting to be able to sit, in some cases walk, and certainly live a lot longer than we previously were not otherwise able to see. In conditions like Rett syndrome, which there are gene therapies and RNA therapies in development, the oral therapy that was approved was specifically to help behavior and quality of life in a complex multi-system measure, looking at many different aspects of the rare genetic disorder and seeing improvements on therapy compared to off oral therapy. So there's different kinds of ways to see improvements, whether it's actual survival to just day-to-day quality of life. And so some of the steps that would be needed is better understanding what are the major issues in ZMYND11 related neurodevelopmental disorders. Is it behavior? Is it speech language? Is it motor? What are the main things that we really need to try to tackle as clinician researchers? How does it change over time without treatment? And then with a treatment, what kind of changes should we expect to see? And so a lot of these disorders have had years of observational data or studying just the natural course of the disorder in affected individuals before an intervention was available. But yes, there's absolutely a lot that can be learned from these other rare genetic disorders that could be applied to ZMYND related NDD.
And I guess last question is, I know that ZMYND11 is very rare. Is there a database that your organization is tracking, and that could we be a part of that?
So I'll answer that. So we are going to have a presentation later on a new gene registry that we've just registered ZMYND11 with, so that we can start tracking all that data.
Thank you so much.
Yeah. And there's a couple in the chat, but I'm going to have Malay go first. She had her hand up.
Hi, I'm Molly. My name's
Okay.
spelled really unique. And my nine-month-old daughter is affected. We got a microarray done, and I'm just curious, her geneticist didn't seem to have any urgency in doing like the whole exome sequencing, just because she kind of said that a lot of the information we may find may be benign, or we don't know anything about certain things that we find. So how useful would it be? But I'm just curious from your perspective, doctor, if maybe it may not be super useful at this moment in time, but maybe as science progresses, would that be useful information to just have on hand?
In general, if someone already has received a genetic diagnosis from one testing technology, I don't necessarily require doing a second testing technology. On a research, likely it's going to show the same thing, namely that that section of DNA is missing that includes the ZMYND11 gene. Now, microarray can sometimes be more accurate in determining the overall size of the deletion, where exome may not actually be able to determine the size. And sometimes it's helpful when thinking about deletion syndromes, or thinking about kind of like if a whole section of DNA is missing, what other genes may be involved that could potentially affect what other health issues may arise, or what may be similarities and differences to expect in your child compared to other children who have different types of genetic changes in the same gene. So I'd rather go back to the geneticist and say, okay, what is the deletion size? Are there other genes involved? And what does this mean for my child, if anything?
And then let's see, in the chat we have, we always heard that gene mutations can't be treated. Is the C-type different with others, which has the possibility to be treated than others because of its own different function? And again, we are going to have a chat later on drug repurposing also for treatments. Did you have anything to add? Yeah.
I guess I want to make it very clear that all genetic disorders are treatable, maybe not curable. And so I think there's a big difference in saying you have a genetic disorder and there is no hope, to say, hey, there's a genetic disorder, we know the cause, and we can best plan for the future and create strategies and potentially new treatments in the future to change the course of disease. And so I think treatment may not be a cure, but there are both behavioral strategies, either drug repurposing or even genetic therapies that could potentially modify someone's underlying genetic information or help support someone's underlying genes to be better functioning. So I certainly want to offer hope and not despair. Yeah, the duplication situation is very interesting and relates to this question about dosing, so gene dosing. A lot of the focus initially was on loss of function, but there is a suggestion that gain of function could also potentially be problematic. Now, in that main 2020 paper, they did end up sorting out individuals who may have had loss of function to individuals who have gain of function, and duplication with gain of ZMYND11 would potentially be related to the problem would actually be too much ZMYND11 rather than the opposite. And so I think it's just very important when conducting research that that we know it's not just all people with ZMYND11 related NDD, but ZMYND11 related NDD due to loss of function or gain of function, because the implications could be very different on a mechanistic level. I don't know if the laboratory ended up calling that a variant of uncertain significance or actually disease causing, but certainly there's ongoing questions if gain of function is really a disease causing or not.
I think it's important too to know that pretty much everything with this gene is going to be driven by the people on this call. So there are not doctors out there necessarily that this is their job. They're not being paid to do it. There is research happening, but a lot of things are going to have to be moved forward by this group. DISA has done an amazing job of getting a lot of these balls rolling, and we'll talk about some of the registries and funding and different things like that later. But we're going to have to be the ones to advocate and to get the information and continue to push funding, research, registries forward.
Do you recommend people see a geneticist? I know most of us get a genetic report. We see maybe a neurodevelopmental specialist and a counseling call.
I'm a neurologist by training, but I think there can be a role in terms of thinking about the different organ systems that may be involved and screening. Really, we just are so early in our understanding of the entire spectrum of disease in ZMYND11 related NDD that to make sure that you've received appropriate genetic counseling and appropriate surveillance going forward, it may be helpful, but ultimately it depends on each person. It's not absolutely necessary, but maybe something worth considering to make sure that you know the ins and outs of your particular variant or gene change and its implication for your loved one. Even if someone has the genetic disorder, and there's this 50% risk of passing it on to children, there's a way to do advanced reproductive planning, or their advanced reproductive technologies that can adjust that risk. And so if an individual affected with ZMYND11 related NDD wants to have children, speaking with a prenatal genetic counselor or their OBGYN to review genetic testing strategies, including the possibility of what's known as pre-implantation genetic testing for monogenic conditions, or PGTM, which uses testing of embryos to determine whether they do or do not have the genetic disorder could be very helpful to better mitigate that risk or controlling the risk of having a child with the condition. So there are strategies now to reduce the risk of having a child with ZMYND11 related NDD.
There was a question, which I have had it in the past as well, about like a window of treatment. Can you talk more on that, and if like with this type of mutation, if there is a type of window?
I don't know. Oftentimes within genetic neurodevelopmental disorders, we think the earlier we treat, the better. And that includes with developmental therapies or non-medication or non-pharmacologic therapies, as well as potentially genetic therapies. However, since this protein is likely active through childhood, potentially into adulthood, I don't know its expression over time. It certainly affects early development, but it could be that it continues expression throughout adulthood. And if there are problem behaviors or issues potentially related to that altered protein expression or protein function, it could be treatable pretty much at any age. So I wouldn't stop. I wouldn't necessarily. I think the window is broader when looking at more than just curative treatments. When looking at treatment strategies, I would take a broad age range approach.